Okay, hello everyone. My name is Janaya Driver, and today I'll be presenting my project. Presenting my project on the conservation of ACE2 binding region of the SARS-CoV-2 spike protein across global isolates and different animal hosts. SARS-CoV-2 uses its spike proteins to enter human and animal cells by attaching to the ACE2 receptor. There is a very specific 17 amino acid region within the receptor binding domain that directly contacts ACE2. Because this interaction is essential for the virus to infect cells, scientists expect this region to stay highly conserved. This means the virus should not tolerate many mutations there. My project mainly focused on testing how conserved that region actually is across different parts of the world. Different animal species as well. My hypothesis was that the ACE2 binding region would remain highly conserved across all samples. And so to explore this, I collected spike protein sequences from the NCBI virus database. For human isolates, I looked at sequences from Australia, Wisconsin, Vietnam, and Argentina. For animal hosts, I included the wolf, cat, bat, and beaver. Using the NCBI multiple sequence alignment tool, I aligned all sequences and compared them to the original Wuhan reference strain. I specifically analyzed amino acid positions 417 to 505, which include the 17 amino acids ACE2 binding motifs, and then calculated percent conservations for each sample. For my results section, my results are summarized really in tables 1 and 2 on the poster. And so for the animal host, all 4 animal species, which is the wolf, cat, bat, and beaver, showed 100% conservation. There were no mutations at all within the ACE2 binding region for any of the animals. For the global human isolates, among the 4 human isolates, which was Australia, Wisconsin, Vietnam, and Vietnam were completely conserved at 100%. Meanwhile, Argentina showed 1 single amino acid substitution, elistine, which was K, changing to therion 9, T, resulting in a 94% conservation. If you look at the alignment figure on the poster, you can visually see that nearly every position is identical across all samples, with that one appearing only in the Argentine isolate. So overall, the data strongly supports my hypothesis. The ACE2 binding region of the SARS-CoV-2 spike protein is highly conserved across both species and geographic locations. This makes biological sense because this region is essential for binding to ACE2 and for viral entry. Mutations here could reduce infectivity, so the virus is less likely to accumulate changes in the part of the genome. The strongest conservation also suggests why SARS-CoV-2 is able to infect multiple species because the ACE2 contacting region remains stable across the host. So for the future directions, I would say for future studies could look at newer variants, model how specific mutations might affect ACE2 binding structurally, or perform laboratory tests to measure changes in binding strength. Thank you for listening, and yeah.